Hello and welcome, everyone. I so appreciate you joining the webinar today. For those of you who attended the annual conference in Baltimore, or for those of you who have downloaded the various presentations from the conference, you might remember that Prosensa was in its quiet period and therefore had limited ability to talk about their program. Prosensa has come to PPMD, and we've been discussing the need for a webinar to further talk about their program in antisense oligonucleotides and how and when we'll see other exons in development. So Holly from PPMD is going to manage the webinar today, and so with that, I'm going to turn the webinar over to Holly. Holly?
Great. Thank you. Welcome to our webinar on the Prosensa exon skipping Duchenne program. I'm going to introduce you to Dr. Giles Campion from Prosensa, where he's the Chief Medical Officer and he's the Senior Vice President of Research and Development. He's going to talk to us today about Prosensa's exon skipping program, and that's going to include a review of their past progress as well as a look to the future. Just a couple of logistical items before I turn it over. So first is please feel free to put questions in the chat box. We will take your questions during the webinar. There will be time at the end to answer those questions. If you have specific questions about you or your child's mutation or particular status, we'll answer most of those offline, but we will answer all the questions we receive. We also will record this webinar, and it will be available from the PPMD website, so you're welcome to go back and listen to it again. So with that, I know you're very interested in hearing from Dr. Campion, so I'm going to go ahead and turn it over to him. Welcome.
Thanks very much, and I'd like to thank Pat and PPMD for the invitation to give this webinar to be able to go through our program. And so I do that right away. As you heard, I'm the Chief Medical Officer and Senior Vice President from Prosensa, and what I'd like to do is, apart from the forward-looking statement that is obligatory as a publicly traded company now, I'd like to give you a little bit of an introduction to Prosensa, because being on the other side of the Atlantic, perhaps not everyone is quite so familiar with us. The company is relatively new. It was founded in 2002. It's located in Leiden, which is a very pretty old established university town in the Netherlands. You can see a picture there on the top right panel. It's full of canals. There are about 85 employees now at Prosensa. We've grown quite rapidly, and our focus is on RNA modulation, where we have our technology platform, with a particular focus on neuromuscular disorders, and of course our chief work is in the area of Duchenne muscular dystrophy. You can see an artist's impression of our labs there, which again are fairly new. We moved in a couple of years ago, and down at the bottom are the employees of Prosensa, all working to support our effort in Duchenne. Just to give you a bit of a background to the timeline involved in exon skipping, and also the intimate way in which we've been working with the patient groups, which I have to say have been absolutely critical in supporting our efforts, both in the very, very early years in Gert-Jan van Ommen's lab. This was in Leiden University in 1998, when he started to explore exon skipping as a potential therapy for DMD. And then as Prosensa was founded in 2002, particularly the Dutch patient project, parent project in the Netherlands, and also PPMD in the early years, and Cure Duchenne also made a significant contribution. And over the years, many other patient organizations have contributed as well. So we've always been very close to the patient organizations. They've been critical for us to be able to get to where we are today. In 2007, the chief scientist working in Gert van Ommen's group, Judith van Doetinchem, joined Prosensa, and of course she brought all that expertise with her, having been involved right from the beginning in exon skipping. In 2009, the first exon skipping trial was published in the New England Journal of Medicine, and that's the year we signed with a collaboration with GlaxoSmithKline, and that was transformative for us because they, in licensing the lead program, we were able to work with them and exploit all the resources that they could bring to bear to the project. But also they gave us additional funding to be able to start development on all the follow-on exons, and I'll be talking a little bit more about that during the course of this presentation. In 2007, we had the second exon skipping trial published in the New England Journal of Medicine, and I emphasize that because the New England Journal of Medicine is the top-rated medical journal in the world, and having articles published there really helps bring publicity to the whole area of Duchenne amongst the people that read it. In 2013, the first positive placebo-controlled trial of exon skipping in DMD was reported. This was by John Krause from GlaxoSmithKline at the Cold Spring Harbor conference. And then, as you may be aware, breakthrough designation was received by GSK from the FDA towards the end of June, and that really designates it as a priority project for the FDA as well, and they will treat it with the highest priority as it comes to, hopefully, to registration. Say a little bit more about Judith's work and the discovery group. Having all those years of experience of exon skipping means that Judith is a real expert in antisense oligonucleotide design and has been working on the follow-on exons and using her experience and the expertise in Prosensa to optimize and find molecules to make sure that they are as good as we can make them. We also, in the company, have a group that is dedicated to the production of early molecules, both for working in the lab and also doing tests in the animals. And then they also have the knowledge how to scale these up and make sure that they are produced to high quality, and then also can help to transfer these molecules into the bigger contract manufacturing organizations that we use to produce these molecules to scale. Another group that I'd like to emphasize is the group that work on biomarkers and bioanalysis. As I will go on to explain, and you're probably aware that the GSK program is extensive. We're doing all the bioanalysis, we're doing all the dystrophin measurements for that trial. And if you can imagine, with over 300 patients in that trial and multiple biopsies, we've really had to work hard to develop a robust and sensitive method which is operator independent, so that we can be confident of our measures of dystrophin. And we're also part of a consortium of expert labs which is working together to be able to standardize the analysis of dystrophin. Well, as you're aware, the approach that we take with exon skipping means that antisense molecule is not enough. We need a whole portfolio of them, and that indeed is what we're striving to do. And 2013 will be an important year for us. The results, as I alluded to, of the first placebo-controlled trial, which was positive, was reported in Cold Spring Harbor. The results of the confirmatory phase 3 trial will be available at the end of this year. As far as the other programs are concerned, we've finished the phase 1/2 trial for exon 44, and we anticipate starting the extension study by the end of the year. And another 2 programs are either already in the clinic or just about to start imminently. So in conclusion, we have 4 programs that are already in the clinic. We have a further 2 programs which are in late preclinical development involving skipping exon 52 and 55. And I know this is a very important theme in the community, that's how to deal with individuals with rare mutations. This has been something that we have been thinking about quite hard over the last couple of years, and have a very high priority program in research called Prospect, which I shall come back to shortly. So now to turn to talk a little bit about drisapersen and why that's important for our follow-on exon strategy. As you're aware, this is a global study that has been conducted by GlaxoSmithKline. There are over 300 patients in more than 50 trial sites in 25 countries, and each of those yellow dots signifies a different trial site. And you might say, well, why is this such a big study? It is unprecedented in the area of rare disease, particularly Duchenne. Well, the thinking has been like this. If you can show the regulators and also the payers that you have definitive evidence of safety and efficacy in a large group of people, then our thought is this will make it much easier to then go back to them with these results and say, now let us find some way together to move the follow-on exons through more quickly. And so that has been an important part of our strategy in accelerating the follow-on exons. The next important question I know you want me to address is what about plans for the US centers? And I want to divide this into two bits. One is to talk a little bit about our natural history study. I did mention this at Baltimore, but I want to emphasize it again and how this is integral to our whole strategy. And then I'll address the therapeutic trials that we're pursuing. So this natural history study is a global study. We're doing it together with GSK, although we're actually handling it here at Prosensa. We aim to enroll 250 patients over 3 years. A number of objectives in this study. First is to characterize the natural history and progression of DMD to help inform the design of future studies. And you may remember back when we first started our program, there was very little known about any clinical endpoints, and it's only relatively recently that so much has been learnt about the 6-minute walk, which is the primary endpoint used in ambulant boys. But still relatively little is known about the younger boys, about those that have lost ambulation. And we've been working with groups, including PPMD, to design new endpoints for the non-ambulant population. One I mentioned in the slide called the PUL, or the performance of upper limb. And we aim to use the natural history study to test that so that we can understand how that changes with disease, not only in the non-ambulants, but also those that are in the transitional phase between when they can still walk, but are soon going to be losing ambulation. And this would allow us to follow something right away, right through the clinical studies and not through the period that individuals move into losing the ability to walk. The second objective here is to capture biomarkers. I think everyone's focused on producing some quick markers that show that drugs are working, so that we don't have to go through long clinical trials to be able to get registration. I think everybody has that as a goal. We do too. We're collecting different types of biomarkers, which are proteins found in the blood and urine, which may be linked to disease progression. And the other element of this is safety. As you'll find out, I'll talk about later, is that some elements of safety profile we have noticed appear not only in those treated but also in those on placebo, something that we weren't aware of, and I think we need to understand a bit more about what the profile is in boys with Duchenne generally, as they don't normally get blood tests. Most importantly, for accelerating our follow-on exons, the natural history study will provide comparative data for the development of the rare target exons for which no formal controlled trials are not feasible. And what do I mean by that? I mean, if you only have very, very few individuals in a country, maybe a handful, then it becomes very difficult to do placebo-controlled trials. And we know from some of the work we've been doing that if you only have 4 or 5 placebos, then it's very unpredictable what result you'll get if you follow them for a year, for example. So our aim is to, rather than to do that and also have to have placebo in every trial, is to be able to use this natural history data, which will be at the same time as we're running our studies, to provide the data that we need to show evidence of safety and efficacy. So we have 3 centres at the moment in the US that we have plans to potentially to increase that. But if you're interested in participating in the study, I've got the link there on the slides, and as you heard, the slides will be available after the webinar. No medication is being tested in this study, and patients can stop participating at any time. But it really will, if we can get this up and running, it really will contribute to the acceleration strategy. We have now some 85 patients in, so we made a good start, but we need to carry on. Moving to the therapeutic trials, Pro44, as I indicated, the phase 1/2 has been completed in Europe. The results will be presented at the World Muscle Society meeting, which is taking place in California in early October. The extension study is anticipated to start in Europe at the end of this year. And then the pivotal study with US sites is anticipated to start towards the end of next year. Just talking a little bit about Pro44, because the question often comes, well, why are you doing your early work in Europe? What we found in the past is that, and this is relevant for this program, was that start in Europe with more limited preclinical safety data. And also the US authorities wanted to see a single-dose human study prior to multiple dose studies. We wanted to do multiple dose studies because if we wanted to look for drug effect, such as dystrophin, a single dose study wouldn't really help. We really needed to do a multiple dose study. So that's why we started in Europe, so we could get going, we wouldn't lose time. And then the long-term study in the US can start once long-term preclinical safety studies are complete. And these studies are anticipated to be ready at the end of 2013. And as I said, this principle explains why our early dose finding studies have tended to start in Europe before the US, but we're working to change that. The next program is that of Pro45, which is targeting exon 45. This is an example of where feedback from the patient organizations have been instrumental in making us think about our study designs. I think one of the aspects of the initial studies we did, both for Pro51, which is now drisapersen, and Pro44, was that there was a phase where you look at different doses, then you'd stop the study, try and analyze the result, and then you'd start the next study. And that has quite a lot of delay in it. And also had the disadvantage of meaning that individuals didn't have continued access to drug. So what we've done with this design was we have now got a single study, so-called seamless design, where we start at different dose levels, but then boys that are on different that start at the lower doses are kept on that dose and then are gradually moved up to the higher dose so that all boys continue on drug for the whole study. And then that all rolls on to what we call a treatment phase, in which additional boys are recruited so that we get a good 48 weeks of treatment. And it will be that treatment phase that will be compared with the natural history study to convince regulators of benefit. So this whole new design has been brought about by our interaction with the patient groups. Now, as I say, what we're trying to do is each time we do a program, we hope to do it quicker. And this is reflected in the design with 53, this has a similar concept of keeping boys on drug, but instead of doing longer-term dosing in the dose escalation phase, we're just doing single PK doses so we can understand how the drug is working in the body. Obviously, this information that we've had on the trials beforehand, and then we end up with the two highest well-tolerated doses that we treat for slightly longer before going into the treatment group. And another bit of feedback that we've received is that it's all very well and good studying these drugs in the ambulant population, but what about the non-ambulant? So this program will also include 10 transitional, 10 non-ambulant boys, and 4 3 to 5-year-olds, so that we can start to get experience with this type of approach in different groups of patients. And this whole program has been discussed extensively with the regulators, so that we are not just doing something that we don't think has a chance of success, that we are putting together programs that we think will be acceptable. I mean, with that caveat, because this reflects to, again, refers to questions that have been posed prior to the webinar, I think, if the results that we get with drisapersen are really outstanding, then I think that allows us to go back to the regulators and say, look, we've got this evidence that this whole process works well. What can we do further to try and accelerate these studies? And these are the data that we have to date. So I think, that there is an opportunity. We found that the regulators, and partially through all the information that you've been giving them, are becoming very receptive to dialogue and to talking about how we can work together on these programs. The other two programs, as I say, advanced preclinical studies are ongoing. We would like to be able to start these programs in the US as well as Europe simultaneously. That's a goal that we have, and we will be scheduling a pre-IND meeting for at least 52 during the coming year, and potentially also for 55. So those are the programs in development, and what this chart does is just show the focus of our current exons, and those blocks underneath signify the exons and the groups of mutations that we're currently addressing with the current portfolio. But the red circle is talking about, well, what about those outside the so-called mutation hotspot area? What are we going to do for those boys? And what I'll show you on the next slide is the frequency of not only of deletions in that group. So you see there are a lot of mutations that occur, a lot of deletions occur, but it only affects a small number of individuals compared with the so-called hotspot, where you from exon 45 to 55. Below that chart is one showing the duplication frequency. So, of course, we're not only concerned about deletions, but also duplications. These are also important. So what do we see as the response to that? Well, what we've been looking at is multi-exon skipping, so that if we can remove a segment of the dystrophin molecule, which we feel is not essential for function, in a larger section of the molecule, then we address a bigger proportion of the population. And these two panels here show the results of two different experiments. One in skipping exon 10 to 18, which will be applicable to about 5% of the Duchenne population. And what these various bars mean, the bars on the left-hand side are marker bars, and then under the T, that is the bar in which treated healthy cells have been treated with this multi-exon skipping technique. and you can see at the bottom there, there's the band, well, at the top there's the band showing the intact 9 to 19 segment, and at the bottom there's a band where the exons 10 to 18 have been skipped. And the next band is the non-treated, and you just see a single unskipped band there. If you go to the next panel, that's another experiment where we've applied this technique to trying to skip exons 10 to 30, and you can see that there's a similar result there with the presence of a skip band. That's the far left band in which exons 9 to 13 have been skipped. And if we're successful with that, that would be applicable to about 13% of individuals with rare mutations. So this is, I must emphasize, this is early discovery, but it's something we're excited about and treating as high priority. Evidence has been seen in the lab and reproduced a number of times. But at this point in time, and again, I'm trying to address some of the questions that are being asked, it's too early to predict the timing of clinical trials, but you can be rest assured it's something that we're putting a lot of resource on at the moment. I want to change tack a little bit and talk a little bit about safety. These were questions that came from PPMD particularly, and one item that you may have heard about is protein in the urine that has been seen in the Drisapersen program. And to try and explain that, I need to just tell you a little bit about how the drug works once it's in the body. Once the drug is injected, it distributes apart from the muscle, which is where it has its action. It's also distributed to the kidneys and concentrates in an area of the kidney called the proximal tubule. This area is also the site where small amounts of protein are reabsorbed in the kidney. Protein that's not filtered, that is filtered through the glomerulus, is reabsorbed there. And it seems that presence of drug in the same location may lead to spilling of small amounts of protein in the urine. This is mild and is reversible on stopping the drug. We've also noticed that in the placebo-controlled study that was recently reported, protein in the urine was also found in those receiving placebo, although it was at a lower frequency. In two cases that you may have heard about, protein loss was more severe and resulted in the drug being stopped. But this, we think, was operating through a different mechanism and wasn't associated with this tubular mechanism. And these boys have recovered on stopping drug. Over 300 boys now have entered the Drisapersen clinical trials, and of these we're not aware of any negative long-term renal consequences. Protein in the urine is easy to detect by a simple urinalysis, and the experience we have to date from this whole program suggests that this is a manageable effect. The next topic I wanted to talk was about pain and discoloration during subcutaneous injection. This medication can be sticky at low temperatures and painful if injected in that state, so it's important that it is brought up to room temperature, and this is something that we do emphasize to the sites participating in the trials. GSK has presented data from the phase 2b study earlier this year. Injection site reactions, and these were mild to moderate in severity, and no patients discontinued treatment. Skin reactions were also reported with placebo, although at a lower frequency than treated groups. And I know some questions have come in as, how do you keep the studies blinded if there's skin reactions? I think the fact that we also see them with placebo helps mitigate that issue. To date, of the whole program, only one subject has discontinued due to pain or fear of pain on injection in the Drisapersen program. And skin creams such as Rapidan or Imla has been used in some individuals. I think the important thing to say about the subcutaneous route is it does offer a lot of flexibility for the patients and offers the possibility for home administration, which would reduce the burden of having to go to hospital all the time for drug administration. want to talk a little bit about the chemistry, because this is another topic that's come up. When the work was going on at an early stage at the LUMC, and Judith van Doetinchem was involved in this, there was a choice of chemistry. I mean, at that stage I could have chosen any of the chemistry that's available right now, including the morpholinos. The choice to go with the 2-O-methyls was based on the fact that there was already quite an extensive experience of these molecules, so that the safety profile was relatively easy to predict. These are 20 to 25 mers in length. They're negatively charged, and that's important because it means they bind to protein, which we feel improves the ability for them to be taken up by muscle, and it also improves the time that they're available in the body. So you don't actually need that much of them compared with some chemistries. So our current dosing in the phase 3 trial is 6 milligrams per kilogram every week. Another important factor in the choice was the availability and the ability to manufacture, and that's been important. GSK now has been able to expand manufacturing so that it is being provided at a scale that means that the whole market will be possible to supply the whole market. As I indicated, the route of administration can either be subcutaneous or intravenous, so it can be flexible there. In terms of the safety, apart from accumulation in kidneys that I talked about and liver, which hasn't proven to be an issue in the clinical program to date, it can also, at least in the preclinical animal models, can interact with pro-inflammatory factors, which may be a factor in some of the injection site reactions, but we haven't seen evidence of systemic inflammation in the program to date in the trials. So, as far as clinical studies are concerned, as I've indicated, we've looked at dose ranges from 0.5 to 9 milligrams per kilogram per week in over 300 patients, 50 sites in 25 countries. And again, from a safety perspective, having studied all those individuals, and some of them have been on drug for up to 4 years now, it means that we're relatively confident about the safety profile that we will see, and that we can develop the appropriate monitoring procedures to make sure that boys come to no harm. As far as detecting the sort of molecular aspect of drug effect, we look at dystrophin in the anterior tibialis muscle, but I think the important thing is that all our program has been done with the accepted regulatory endpoint, which is the 6-minute walk test, and that again addresses one of the questions: why haven't we gone for accelerated approval? Well, why hasn't GSK gone? It's because we don't need to, because accelerated approval is only if you're trying to go the surrogate marker approach, which is getting approved on dystrophin. We don't need to do that because we've done all our studies on the functional endpoint. And after all, what's more in boys is whether they know they've got dystrophin in the muscles or whether they can actually walk further. So I think that's the way we've approached this. Steps that we've taken to try and speed up development. I talked about the integrated clinical designs. One thing we've been able to talk to the regulators about, since these molecules are all very similar, they have the same chemical backbone, we are seeing a lot of similarity in the way that they behave in the body. And so we've been talking about the necessity of repeating all these time-consuming and expensive preclinical studies, and we've been able to successfully negotiate reducing those. We've talked about avoiding placebo. That allows us, particularly for the rare mutations, we can maximize the use of the boys that are available and not have to use placebo on some of them. We are working hard, as I said, on looking at other markers of effect. Again, using the 6-minute walk test does mean that you have to treat boys for a while to see that effect. I think all of us would like to see measures either dystrophin, maybe in combination with imaging or other markers of biochemistry as a way to speed up development. And we also are working hard to speed up the manufacturing process, which again is something that people don't talk about a lot, but it consumes critical to be assured of a certain drug supply. And the way we do that is somewhat similar to the way we're approaching the preclinical safety side of things. By building up a lot of experience in making these molecules, we can start to predict what we need to do in terms of assuring stability, in terms of assuring the quality of our manufacturing process. And so the amount we have to do for subsequent exons, we hope will decline. And again, that's something we're discussing with the regulators. A big event for us, and was also important for our whole approach to Duchenne, was our public listing at the end of June. This gave us access to public funds, which means that we have a really good safety buffer now to make sure that these programs can continue unimpeded. One thing I said in Baltimore, and I'd like to repeat again, I mean, being on the road for 2 weeks trying to convince investors to buy your shares, we decided that the patient groups and PPMD in particular have done a fantastic job in raising disease awareness, because every single investor we went to talk to knew all about Duchenne. So that's a fantastic job that you've done as a community and need to be proud of. We are committed to working with patients. We highly value our frequent contact with patient organizations. Have played a pivotal role since the very beginning, and we consult to know what is important to patients and their families. I gave them the example of the way in which discussions have changed our approach to designing studies for the follow-on exons. We have the largest exon skipping portfolio for DMD in the clinic. As I said, we've taken steps to be able to include some of the other patient groups, the non-ambulatory and the younger boys, in our current programs. We have a seamless design for 45 and 53. The extension study for 44 is anticipated to start this year, and we're undertaking many efforts to speed up development. If you do have any questions, we have Claire Layton, who's actually sitting beside me as I give this talk. She's the manager of patient group relations, and she's incredibly diligent in terms of replying to every question that we get on a personal basis. And some of the individual questions on individual mutations, she has formulated responses to, and they will be sent to you after this webinar. So in summary, Prosensa is committed to developing novel medicines for DMD. We have deep expertise in oligonucleotide design, manufacturing, preclinical and clinical development. We form partnerships with scientists, physicians, patients and patient groups to help develop and understand DMD and our drugs. A partnership with GSK to globalize reach and bring depth to the portfolio. And I do want to emphasize that. As a small company, there's no way we could do, for example, the submission program that they have planned for drisapersen. We could not submit in a simultaneous fashion to multiple countries to make sure that boys all over the world can get access as quickly as possible to the drug. We don't have the resources to go and talk to all the payers to understand what they want to be able to reimburse the drug. We wouldn't be able to put in the programs they can to make sure that of speedy access. So we value that partnership greatly. We have 6 products now in development I've been through. With more in research, we're very excited about our multi-skipping program, and we like to think of ourselves as the 3 Ps: Prosensa, Pioneer, and Partner. And I just want to emphasize the individuals that are working very hard to ensure that boys with Duchenne can at least have some hope for the future, that we're doing everything we can to provide a solution for this disease. And up there, apart from myself, is Hans Schikan, our CEO, and again, all the employees. So thank you very much for your attention, and I guess we're ready to take some questions.
Excellent. Thank you very much, Giles. That was very helpful. So I'm going to read off some of the questions that we've received. We have, first of all, a large number of questions about timelines for the United, as well as some questions about inclusion criteria and study sites. So I know that's a lot to sort of add into one question, but perhaps you could briefly review the timelines that you showed before, and then maybe what I could suggest is that PPMD could work with Prosensa to develop a very small grid just based on the information today that we could post on our site for folks.
Yes. Well, what I said was that just going through them in sequence. I guess the first program I talked about was Pro44. I indicated that we were aiming to start up the extension study by the end of the year, and that we would be starting the pivotal study, confirmatory study, by the end of the 2014, and we'd be hopeful to include US sites in that. So that's the current timeline for 44. I must always add a caveat about these things. Timings in drug development are not a fixed item. They are subject to change, but we all work as hard as we can to make sure that we meet these. With Pro 45, we are starting we're right in the middle of doing the dose ranging study now. We will hope to be able to start that in the US in the coming year. And with Pro 52 and 55, again, we will aim to have pre-IND meetings with those in 2014, so that we can start either in that year or in the following year, depending on progress, because those are a little further back. As far as sites, we are. We have the advantage, of course, that GSK has been working in the United States with its Drive a Person program, so we'll be talking to them about the availability of sites there. We're also in contact with organisations such as Synergy that have sites. So we haven't made any decisions about that, but those decisions normally go up when the trial is posted in clinicaltrials.gov. As far as the inclusion criteria are concerned, this will vary according to the different patient population. As I say, although the treatment phase or the pivotal phase of these follow-on programs is in the ambulant population, we also will be including those with transitional disease, so just at the point where they're beginning to lose ambulation, and those that are non-ambulant. And the key thing about the ambulant population is, although a number of the trials are looking at those with relatively high levels of performance, for our trials we're looking at individuals that can walk for the ambulant population more than 230 meters. We do that because we want to ensure that we still have enough boys able to complete the test after 48 weeks. And we've had to discuss that extensively with the investigators we're working with, so that we can have the balance of both being able to recruit enough subjects, because recruitment is an issue with these rarer mutations, but also have something that we think will work as an endpoint.
Excellent. Okay. Thank you. There was actually a follow-up. So would you just briefly remind us about the timeline for 53?
53, yes.
Lots of questions about 53.
Okay. Well, 53, as I said, we are just about to go into the clinic, so we would be announcing that we'll be starting the clinic quite soon. And the treatment phase is anticipated to start in the US in 2014.
Excellent. All right, and I will remind everyone again that these slides will be up on the PPMD website, so you'll be able to see them again. And like I said, maybe perhaps we could put them into a grid or some sort of similar visual so that you could find it easily. So thank you. And I will just reiterate that we are putting Prosensa a bit on the spot here, and that these times are, of course, subject to a lot of things outside of their control. But thank you for doing that. So I have a couple of questions related to, let's see, I'm going to start with multi-exon skipping. So the first one is, would you talk a bit about the expected outcome? Would you expect the outcome of multiple exon skipping to be similar to what you're seeing with exon 51, for example, or would you expect it to be maybe not as effective in terms of benefit, or do you not know yet?
Well, it's early to say at the moment. As I say, the work that's ongoing now is in patient cells, so it's always difficult to predict exactly what will happen. But I mean, as I say, Judith van Doetinchem has got a huge amount of experience now in trying to develop effective drugs in this area, and we're picking areas of the dystrophin molecule where we don't anticipate that removing doing multiple exon skipping will have a major influence on its stability or ability to function as a structural and signaling protein. So that's what I'd say to that.
Okay. And there was a perhaps related question about the feasibility of skipping the actin binding region.
Yes. Well, as you know, there are key elements of the dystrophin molecule where it interacts with other molecules within the muscle where you tend to avoid. So I think, you know, what we're trying to do is deal with the things that are technically the easiest before we move on to things that are more difficult. So we try and pick the things that we think have a best chance of technical success and move those forward first.
We also have a couple of questions about whether children with nonsense mutations or premature stops would benefit potentially from exon skipping.
Yes, they can do if by skipping the exon you don't create an outer frame transcript, and if people have individual questions about the mutations, as I say, I would invite them to send those questions in, and we can deal with them on an individual basis, because it does depend on where that nonsense mutation is, how big it is, and in terms of whether it might interfere with the binding of any of our molecules so that it wouldn't work. So that's something that we can answer on an individual basis.
There are a couple of questions about the natural history study. So the first one is a question about why you all are engaged in a natural history study, why it's important to have one when there are some academic groups who are doing natural history studies as well.
Yes, well, it's the way that we're trying to not have to use placebo. If we're not using placebo, then we need to have boys in that natural history study that match those individuals in our treatment protocols. And if we do the natural history, we can make sure that they've got a greater chance of matching. So that's why we do it. And the other thing is that we do collect information that the other academic groups may not be so interested in. For example, the whole story about protein in the urine. I said we were surprised to find that some of the individuals on placebo also have protein in their urine, so we need to be able to understand these things so that we can really understand the risk-benefit of drugs that we develop.
Mm-hmm. And there's another question about the natural history study, and that was wondering what sort of procedures are required, specifically biopsy, and some idea how long it would last.
Yeah, we don't take any biopsies, and there are just a limited number of blood tests. Part of the objective, as I said, was to try and understand about what biomarkers may be important for us to measure. There are 7 visits over a 3-year period, but as I said, participating in the study is entirely at the patient's discretion. If they want to leave it at any time, they can. And of course, if they come into a therapeutic, offered as a therapeutic trial, then we wouldn't expect them to stay on the natural history study. But it does allow us to collect valuable information that helps, the whole development of our drug platform.
I'm firing these questions to you very quickly. There's some more. So one of the questions was whether you are potentially looking at any sites in Canada. So are you thinking North America, or are you looking in the United States?
Yeah, no, absolutely. Canada is in our list.
Okay, great. What about, would it be possible for people from the United States to join a European clinical site?
Well, the one thing I would say is that we don't have any influence over people participating in studies. I mean, that is really up to the investigator, and the person who wants to participate. So if they want to do that, and they can see on the clinicaltrial.gov where the participating centres are, they can write to the centre and ask.
Okay, thank you. Let's see. Sorry, I'm looking through the questions. There are a couple more that just came in. There are some specific questions about the specific trials. So, for instance, there's one that just came up about how many injections patients receive each time. Some of those might be best addressed offline, but are there general questions or general responses that you could provide about whether the exons outside of 51 will be similar in terms of number of injections and length of time?
Yeah, I mean, that's why we do dose-response studies, because we want to understand, by doing the dose response, what the best dose is. And obviously, if it's 3 milligrams per kilogram, then you have less drug to inject than if it's 6 milligrams or higher. So it's not a question I can answer right now until we finish those dose-ranging studies. But generally, a single injection is adequate.
Good. Okay. Well, again, lots of questions coming in very specifically about which of the trials are potentially going to include non-ambulatory and very young children. And I think that those are things that we can, the community can look at in more detail by reviewing this webinar, and perhaps could be put in a summary table. So I'm not going to ask these very specific questions about.
Yeah. Well, I can answer in general. I mean, it is now our general principle for all studies so each of our programs will involve those individuals. I mean, we want to develop drugs not just for the ambulant population, but those that can be applied to all individuals with Duchenne, because the principle of exon skipping should be valid.
Excellent, thank you. And then the last sort of group of questions we have are lots of questions about the timeline for regulatory approval. I know you can't speak very specifically to that, but I wonder if there's any comments you can make besides what you've already made about the anticipated timeline specifically for 51.
I mean, that's really a question for GSK, but I can say a few things that have already been said publicly. I mean, firstly, I think it's great news that the Drisapersen received the breakthrough therapy designation, because that ensures that it will get top-level attention from the FDA through the submission and approval process. And I know that they will work incredibly hard to make the review process, assuming a file is submitted, that is as efficient as possible. So I think that's really good news. And I think the other good point about it is, as I said, that the approval will not be sought on a conditional basis. This is a full NDA, a full approval that will be sought.
Excellent. Thank you. Okay. Well, I think this is actually actually a good time to stop with questions. I know there are a couple more still coming in. Just as a reminder, all of the questions that have been asked that have not been answered will be addressed. Some of those will be addressed by PPMD. They will as well be sent to Prosensa, and some of those I suspect will then be passed on to GSK, so the appropriate group answers the questions so that you all get the most accurate responses. The webinar is recorded, and you will have access to that recording as well on PPMD's website. So this will be available as a community resource. And, of course, as Giles provided to you, you have access to him as well as Claire. So there are people who are very interested in addressing your questions and concerns moving forward. So I'm going to go ahead and give you a chance if you have a closing remark, or would you like me to close?
I'm happy to. I just want to reiterate my thanks to everybody out there that without the support of the patient groups, we certainly wouldn't be where we are now. And maintaining those close relationships are very important. And also, I should really like to acknowledge our colleagues in the LAMC, because that's where the technology came from. We still have a very close working relationship with them, and that has also been key to us being able to make so much progress.
Great. Thank you so much. Okay. Well, we do appreciate Prosensa's commitment to Duchenne and addressing both the more common and the rare exons. Just personally, it was very interesting and encouraging to hear about efforts to modify trial designs so that they're faster and more acceptable, but also more inclusive. So I think the community is definitely has a lot to be excited about. I'm going to go ahead and close the webinar here, thanking again all of you folks who have joined us, and we will have this available for you on our webinar.